I wanted to say thank you to Selena for inviting me, and thank you to Gina and the rest of the organising committee. This has been really wonderful. I have learned so much. And so I wanted to talk to you about cycads. There's been a lot of talks about angiosperms, and so this is a gymnosperm. And cycads are often depicted alongside the dinosaurs. It's really common, and so what we see is that we have like a vista of dinosaurs, and there's always a cycad plant in there. And so over here you can see a cycad. You know, and even as kids, we draw palm-like plants, which are really cycads, next to dinosaurs. So it's so ingrained in our minds. And the reason for this is because cycads are ancient plants. The fossil record begins in the Permian, so about 270 million years ago. And at this time, during the Jurassic, or after that, during the Jurassic and Cretaceous, they were really common. They were distributed throughout the world. They're even found in Antarctica and in the UK, where they don't occur now. And they have managed to survive until today. And out of all of the seed plant groups, such as angiosperms and conifers, ginkgo, they are the oldest living group of seed plants. So today we have about 350 species of cycads. They occur in tropical and subtropical regions of the world. We are still discovering more species as we keep exploring the cycads. And so what we see is that we have this picture of cycads as really ancient plants that have managed to survive until today. And when we look at the fossils, often we call the living plants living fossils because the fossils look almost identical to the living plants. There's a trunk, there's a crown of leaves, and a cone. So what I have been really wondering about through my research is how have cycads managed to persist for over 270 million years? What are the mechanisms that have allowed them to stay on this planet when the dinosaurs have died out? And so I use a range of tools, and I'm going to talk about those today, and all of those different approaches that I've tried to use to answer this question. And so one of the first studies that I did on cycads was to do a dated phylogeny. And what I did was basically I got a phylogeny and I used the gene phytochrome. Baronda was talking about using phytochromes yesterday. In this case, I used phytochromes to basically do a family tree. And what I found, I also used fossils. You can see these stars. The phylogeny is basically stretched out through time, and so the fossils help constrain those time points across the phylogeny. And what I found was that each of the genera are pretty old, and so you can see that by the beginning of each of these colours. So for example, the green colour, the red colour, the purple colour. The origin of the genera can be pretty old, from the Jurassic all the way to the Oligocene. On the other hand, all of the species are really young. And I trained as a paleobotanist first, and so when we talk about young, 12 million years is actually really young. And so we think of this time scale as really young, and things like Permian is really old. And so for me, this was remarkable. These cycads that were supposed to be really old are actually really young, and they started radiating in the last 12 million years. So basically where each of these brooms kind of start their diversification. And the other thing that I found really remarkable was that this radiation was synchronous, and so it occurred in multiple regions throughout the world at the same time. So for example, you can see the purple, which is a bunch of African cycads. They start to radiate at the same time as South and Central American cycads, and also Australian and Asian cycads. And so this occurred during the mid-Miocene. So if we have a look at what the planet looked like in the mid-Miocene, this looks really similar to the world that we know today. The oceans, the continents, were in pretty much the same configuration. And what this tells us is that the oceanographic patterns were the same as today, pretty much. And so it indicates that we had a pretty modern climate. And what I mean by a modern climate is a relatively cool climate. So if we look at proxies for temperature, what we see in the mid-Miocene about 15 million years ago is this climatic peak in terms of temperature. As we go down through time, we start to see really cool environments. And so this is really what marks modern conditions, is these cool conditions. So coincident with that, we have the establishment of ice sheets, we have cool summers, and we also have seasonality established in the last 12 million years. And so it seems like cycad diversification is happened at the same time as some kind of climatic event. And that's because it happened all around the world at the same time. I've tried to think of other mechanisms that could have caused that, and really the only mechanism that I could think of that could cause such like a global diversification is climate. And so one of the groups that diversified in the Miocene is the genus Cycas. Today, it's the most widely distributed group. It occurs in the north of Australia, also in the Pacific Islands, all the way into Asia, India, Madagascar and Africa, and even all the way into Japan. And you may know that species as the sago palm, so Cycas revoluta. It's grown everywhere. I've seen it on sale in Whole Foods and in IKEA. Many of you probably have it in your gardens. The group is really easy to identify by several morphological characters. And so what I have been curious about is what are the relationships among these various species of Cycas. And so a paper that I was a co-author on earlier this year tried to generate a phylogeny of this genus using Sanger sequencing. And we had about 10,000, almost 10,000 base pairs. So this is a pretty large data set for phylogenetics. And unfortunately, using just the standard markers that we use for phylogeny, we really don't get a lot of support for the relationships. So every single green branch is a branch in this backbone that we don't get strong statistical support for. We also don't get a lot of support at many of these sort of tip branches, and I didn't even colour all of those because it just would be mostly green. And so because of this, we don't really have a good understanding of how these species evolved. So this brought me to using phylogenomics. And so I started to explore what kind of genomic tools I could use in the Cycads. We don't have many genomic tools in the Cycads, unfortunately. So the genomes are really big. The smallest genome, and that's in this genus, is 14 gigabases. So this is enormous. We don't have a full genome of Cycads, and we have, there are a few transcriptomes out there, but very few, and they're not annotated. So that wasn't useful either. So then I decided to go down the line of RADseq. And really simply, what RADseq is we get restriction enzymes, we chop up the DNA into fragments using those restriction enzymes, and then we just select a subset of those fragments. And typically the subset is about 200 to 500 base pairs. So it also makes the huge genome much more manageable. When I sent all my data away for all of these species, I got 85 gigabytes of data. So it's a huge, I've never dealt with anything that big before. And so then I ended up with a matrix of 1 million base pairs, and this just blew me away. We were working with 10,000 base pairs before. And so this is a phylogram, and so what you can see is the branch length is proportional to the molecular evolution. We can see that the group Cycas forms a very well-supported group. So 100 is the best value that we can get. What I've done here is to transform that phylogram, so the branch lengths, into equal branch lengths, which we call a cladogram. And what I was also super excited about was to see all of these really great support values. So from having a tree with very little support, we've gone to lots and lots of hundreds, and almost all of the branches are supported in this phylogeny. So this was amazing for me. So when I started to look more closely at these groups, I found that most of these groups have very strong geographic patterning. So for example, you can see there's a Southeast Asian and Asian group, there's a Philippine group. This group over here is a little bit weird, and the reason is that the seeds of these group actually float on water, and so they're able to disperse really well across the Pacific and also into Madagascar. Most of the other groups, the seeds basically just fall down. And I think Jill mentioned that the Bucera has the most boring seed dispersal. I think this is even more boring because at least you have a fruit. We don't even have a fruit. The seed it just falls down and that's it. And so one of the most exciting things here was, as you can tell from my accent, I'm from Australia, and so I was really interested to see the patterns in the Australian cycads, and I found they were a really well-supported group, so all of them clustered together, and even within Australia we get really strong geographic patterning as well. And so for those of you familiar with Australia, we have the Northern Territory, so like the top end cycads, and the Western Australian cycads forming a group. And then the Queensland cycads, so those on the east coast, they form a separate group as well. And so this really strong geographic pattern really indicated that there's really localised, or what I'm sort of thinking about like in situ diversification, and that we have very little dispersal once these species originate. And so I really wanted to look a little bit more closely at one of these species to see the role of geography below species level. And so what I've done is to look at this species, Cycas calcicola, that I've marked with a star here. And so this occurs in the top end of Australia. Here we are in Australia, and the map that I'm showing is the inset of this over here. And so this is a project that my PhD student has been working on. So a couple of years ago, we went out into the field, into the heat and the humidity and the mosquitoes of the Australian outback. We collected Cycas calcicola and a few other species, but Cycas calcicola is in this light blue colour. We collected it in the Litchfield National Park area, and we drove 3 hours south down to the Katherine area and collected it down there. We were also able to get Botanic Garden samples from the Daly River area and also Spirit Hills, which is actually way off this map. And just to show you what it looks like, to have some nice field shots, this is in the Litchfield National Park area. You can see the cycads have this kind of these sort of grey greenish colour, and that's because they're really, really hairy. Another sort of thing that we deal with in collecting cycads is this really long grass, and oftentimes this species occurs on limestone. You can see the calcicola name, and so there's also like sinkholes as well, and we deal with snakes and just everything really. So that was the Litchfield population. We also have a population from the Katherine region, and I love this photo because it shows you that like real iconic red outback of Australia. This was actually on a university cattle farm, and we just drove up and said, "I heard you have cycads," and they said, "Sure, follow us to the back." And here we have a whole bunch of cycads peeking out through the grass. So we collected cycads in the Katherine region too. And so my student James also used RADseq as well for these data, and he did a PCA on the SNP data. And just to sort of walk you through it, we have each individual dot represents a plant, and each colour and circle represents a whole population. And so we collected, you can see, 3 populations in this region. And so when we mark out where these plants are from, what region, we can see there is a strong geographical pattern. All of the Katherine plants cluster together, the Litchfield plants cluster together here. There's a few Daly River plants and the Spirit Hills plants as well. So there's really we see a lot of genetically distinct groups in these data. When we look at the structure plot, structure suggests that there are 2 clusters in the data, and so the 2 clusters basically correspond to a predominantly yellow group and a red group. And so each bar represents one plant or one individual. And so the Litchfield plants cluster with the Daly River and the Spirit Hills plants, and the Katherine plants, they all cluster together as one unit. And so it looks like geographic distance is important in shaping the genetic diversity of cycads. And so just to sort of summarise and bring it back to my first question, how have cycads managed to persist for so long? It looks like geography has played a really important role in generating the diversity of cycads, both at the below species level and also at the sort of family level and between different groups as well, groups of cycads as well. And that's because there's very little dispersal of the seeds and also the insect pollinators between these regions. And it's likely that the trigger for the diversification of cycads was climate. And so at about 12 million years ago, we get the establishment of seasonality. And what I think happened is that we get these very regular reproductive cycles that help things like genetic drift separate these different species or different populations eventually into different species. And with that, these are my acknowledgements, and thank you.